Okay, so we are recording. So thank you again to the 5 of you that have logged on so far. If at any time you have a question or comment for Gary and Greg, you may feel free to raise hand icon. If anybody has any questions about where that is located, you can send me a private chat. Feel free to use the raise hand icon, which bumps you to the top of the participant list. And at that point, I can call on you individually to respond or ask your question. You may also feel welcome to use the Q&A pod to submit specific questions that you have, and I can monitor that and find a good time to interject those questions for Gary and Greg. Other than that, there's a few reaction icons, the applause, the thumbs up. You may also use those as a nonverbal way of communicating with Gary and Greg throughout the presentation. So with all of that said, I'd like to welcome Mr. Greg Krug and Gary Klinger, President and Operations Manager of Lampire Biological Laboratories, with a few locations around Pennsylvania. They're going to kick off the Fall 2020 Connect to Industry webinar series for us here this afternoon with, again, I underscore the word wonderful presentation about their really, really neat company. They just have such a unique and interesting operation that if any of you question how do I present and incorporate biotechnology in agriculture to my classes or to my students, well, ladies and gentlemen, you're about to see that in action. Lots and lots and lots of crossover and unique ways of applied sciences working in the biopharmaceutical and biological laboratory space. And Gary and Greg are very, very well entrenched and integrated into the Pennsylvania education system, as well as workforce development, which they may touch on at some point. So with that, I don't want to take any more time away from them. Gary and Greg, it is all yours.
Don, thank you very much. And thank you, teachers, for being here today, but also so what you're doing for us. We're very grateful. We appreciate you creating that future workforce for the life science industry. The agricultural industry is much needed, and we appreciate your efforts. And we hope today that we can contribute a little knowledge to a different niche of the business side of things in the agricultural and life science industry, bringing science and technology together. I'm Greg Krug, President, as you heard, of Lampire Biologicals. This is Gary Klinger. Gary has been with the company for over almost 25 years. I was originally a founder of the company 43 years ago. The company, has grown in a lot of different directions over the years. When we first started the company, our original product was produced from sheep and horses. To produce a product that was a agricultural product, believe it or not, that was used to make biological plates. We would go around to the farmers and we would pay them to let us bleed their animals. And we would collect the blood without using anticoagulants, any chemical means, and develop the mechanical means to separate out the blood from the clotting agents so that we would have a whole blood product that could be used to grow this bacteria on the biological plates. Today, we have grown and we're over 130 employees. We have 5 facilities throughout the state of Pennsylvania. Our clientele is very diverse. It's the major pharma and diagnostic companies, as well as the biotech and life science industry, expanding into the food industry, ecological, cosmetology, and other industries as well. So it's an area that, in other words, has many interactions of the need for our products. And a lot of them are coming from animal-based products, some of them not being where you might typically think they would come from, such as fish or lobsters, as well as some exotic animals like emus that are used in agriculture today, as well as llamas, camels we've had, and we've dealt in other areas of pretty unique features of animals that bring those products, their specificity of a certain need to a certain type of desire in the life sciences. We've expanded our facilities to 5 locations throughout the state of Pennsylvania. We're located in Bucks County. I'll bring it up here so you can get a better look. We're located in Bucks County, as well as in Lehigh County and Bedford County. The facilities that we have throughout the state are either laboratories or animal facilities or a combination of both. We work with animals, in other words, from small laboratories, size animals, your traditional mice, rats, guinea pigs, and such, all the way to a wide variety of your large domestic farm animals, as well as the exotics that we talked about. Our lab facilities are set up for clean rooms. We work a lot in cell culture. Cell culture products you'll hear a little bit about are some of our products, but we also do antibody production, where we're doing monoclonal, polyclonal antibody work in those facilities. We do purification, we do contract manufacturing in these lab facilities, and they're very diverse. The workforce we'll get into here in a little while is very specialized to meet the needs at each one of these facilities. We ship product worldwide. Our clients have a far reach. We talked about some of the types of companies we deal with. In addition to those companies, we also dealing with companies such as the military, the government, CDC, FDA, USDA. We've had products go up into space. We've worked with NASA. The worldwide product needs, you might be surprised if some of the companies, in other words, are located in parts of Europe or Asia, but they have manufacturing needs here as well, and vice versa. The manufacturing needs are overseas. So redundancy and being able to supply product to be able to be, it's a perishable product for the most part that we're making. So being able to ship that around the world and deliver it on-time delivery to certain specifications can be a very complex nature. You really have to work through the customs. It can be very easily, if you don't have everything laid out specifically, getting those products to the different parts of the world is extremely tough. We're going to talk a little bit about quality here. And this is a part that we discuss and that we go over, that we feel is really, could be a curriculum, a course in itself at these levels. You can see some of our certifications. We have the ISO certification, ALAC. ALAC is a volunteer-like ISO certification. It's an animal certification. If you're not familiar with it, it's a higher standard than what's required by USDA. You have to actually pay for the certification, and you're audited by a group of veterinarians every 3 years. Due to our diversification, multiple facilities, we usually have a group of vets that are representing ALAC to stay with us for about a week. The FDA registrations, we have both the traditional medical device as well as the human blood establishment license. USDA, we have both a dealer's license and a research license, and we work with OLA. These are anybody who's getting grant money from the NIH has to work with a facility that's OLA certified. What we're real proud of is our EU classifications here. You'll see the numbers start at 001, go all the way up to 005. We were the first in the state. We had all our facilities done, and this is to be able to ship product directly into the EU. The certifications that we tried to get our technicians on is an ALAS certification. There's different tests that the students can take to meet these classifications. We reward our employees for this. If they get their first level, as soon as they achieve the first level accreditation, they get an automatic $1 an hour raise and a $100 bonus. And there's several levels that they can go to. We had one employee this year that reached 2 levels in 9 months. So they got two raises as well as the bonuses.
And ALAS is the American Association for Laboratory Animal Scientists, and it pertains to both small animal and large animal to some degree.
Right.
So it is good as people go through your types of programs that this is a certification that's out there. If they really love handling animals and working with animals, you know, this is something that anyone can go and take. It's not necessarily easy. They will have to put time into it. It is coursework. It is a lot of knowledge that goes into it, but it is a benefit to personnel looking to get into our industry and handling animals.
Right. And there's a lot of support online. You can join this. You can have your organizations, in other words, whether the school or through the counties, you can join the ALAS has groups that there's a national and then there's local branches, and they will supply you all different types of teachings information. You know, whether it's video, slides, literature. There's a lot of support there that you can draw from. The other part here is you see our ALAC letters. We're showing you several years of our letters. We're very proud of these because they don't typically come out this gracious, but you can see some of the highlight areas of the comments they've given us about our facilities. So we want to talk a little bit about, in other words, our customer focus. It's a very technical business, but our workforce is very diverse to meet the needs of that technical expertise. We have everything from PhD scientists, veterinarians, all the way down to students who we have as senior challenge students or students that are shadowing, coming out of their classrooms directly into our lab facilities, being trained and working side by side with very key scientists or veterinarians, and everything in between, BS degree, MS degrees, high school graduates. So it's a very diverse group. And we do a lot of training and retraining on these. The technology is evolving, but a lot of the basic training that you might take for advantage on a typical farm facility, in other words, is in play at a base level, but then expanded upon. Basis is that we're talking about is that we're required to do our feed testing, water testing analysis, environmental testing of the room, certification of all of our equipment. Again, a lot of these sound like quality issues, and it's very standard of what you're seeing in our quality issues to be in the food industry and other industries as well. And that's why we really encourage you to put into your curriculums at least something about quality, because it's something that might excite some of the students and take them, in other words, into other industries if they find out that the industry that they chose was not the exact one that they wanted to be into, or not getting the opportunity that they want in the areas that they want to live. It's a great way to grow and have a very strong background. Tying into, education and developing the workforce has been something that's been very passionate to me. We actually start with youth at a very young age. We'll start with preschool, kindergarten students, bring them and their parents onto the facility to see the animals, to see how the situation is handled, how positive the environment is for these animals. We feel that, a lot of times when people hear that you're working with animals in science, that it's an animal activist issue. It's an issue that animals are being mistreated or suffering. And a lot of our animals are animals that have returned from the farms. For example, we get outdated dairy cows, meaning a dairy cow that's still healthy, just not a good milk producer, or a beef cow that might have been, a good breeder, but no longer able to produce calves. We have situations where we have horses, polo ponies that no longer are able to be ride hard, but still have their life to live out, and so on and so forth with many different species. So we bring these animals onto our farms and they become blood donors. And we work with them to have this type of environment. Then we work with students, in other words, all through the levels. Again, we do internships, we work with the schools. We actually go into schools and sit as advisors on a lot of different committees. We've been involved in different situations where we develop the programs. Out in Bedford County, where one of our facilities are, we were able to help get them a $10 million grant, put an addition onto the school, and put an ag biotech curriculum together. The growth in other areas, new curriculums are happening in Upper Buck Tech School. We've been involved in their program. They've just redone their labs, and we're hoping to be able to supply them a lot of the equipment that they might need and do other connections with them. I sit on several workforce development boards and economic development boards, again, to tie industry into these opportunities with education and cement the relationship. There's an organization called the Pennsylvania Drug Discovery Institute. They offer free chemicals and glassware that they have in surplus that came in from different pharmaceutical companies back to the academic school systems so that they can take advantage of these situations, as well as offering mentorship and educational opportunities, lectures and such by some of the staff. So we're going to get into a little bit about some of our products so you can understand a little bit about what we do. So we told you about how the company started with the original defibrinated sheep blood and horse blood. Those animal blood products are still a main purpose of Lampire. We supply them a lot of different applications. They're used as calibration controls in hematology machines. They're used as testing for coagulation, how fast your blood clots in the emergency room, and so on and so forth. We also work with human blood products, as well as a very strong correlation with the food industry. We're able to collect animals, byproducts from the abattoirs. So we'll go to the abattoirs, for example, and collect chicken combs. From the chicken comb, extraction takes place of hyaluronic acid, which is then later injected into the joints, particularly the knee joint, as a lubricant to help in downplaying the knee replacement and extending the time before surgery is needed. We'll do other things with cow hides, where we take off the connective tissue layer and the hair layer, and we have corium. From the corium, they make collagen that can be used as wraps for burn victims or in cosmetology or in other types of reproduction. We produce different types of purified proteins and related agents. These related products are dealing with the diagnostic industry for the most part. They go into making either blockers or buffers that we'll use. And again, many of them come from animal sources, like BSA, bovine serum albumin. The bovine serum albumin is used in many, many different pharmaceutical and diagnostic products around the world. And we supply sources both domestically and from overseas in New Zealand. Again, this has a lot to do with disease control and a belt and suspenders to have our clients have secondary sources. The immunoglobulins, secondary antibodies, even our lobster hemocyanin, these are products that have been developed at Lampire here very cost effectively to help in the diagnostic and the making of antibodies. We're very, very proud of some of the work that's going on in these arenas. We were very early on in the establishment of some of these products and services that were in the very early stages of, for example, the pregnancy test that women can now do at home. We have specialty products and services in which we do all different types of work. We have a cell culture media division, all that you're hearing right now with the COVID-19. We supply the viral transport media as well as other medias to grow different types of cultures and do different work. We also have a bag that we developed that we actually manufacture ourselves. That's a permeable bag for cell culture to grow cells in. The bag breathes sterilely, where in other situations, the traditional cultures might be put into what's called T-flask or roller bottles where they have to be gassed with the appropriate amount of oxygen, CO2. Our bag can be put directly into a CO2 incubator and just breathe. we talked a little bit about antibody production. We're going to go a little bit more and show you how these antibodies are used. But an antibody is when you get inoculated for a tetanus shot, you build antibodies to tetanus. So what we do is we take and we inoculate different animals against, could be many different things as you see here. It could be components of different species and different material. It could be a peptide, it could be a protein, it might be a haptene, a drug that you're building antibodies to. If we use the example of the haptene, maybe we're developing a drug of abuse test to test somebody for cocaine or something to that initiative. And these different species that you see here have different attributes that lend themselves to produce different antibodies. The chicken, for example, versus the mammalian, the avian versus mammalian, is producing a chicken anti-Y antibody versus a chicken IgG, versus a rabbit IgG. And this can be very significant in developing different types of tests, both for humans as well as animals. Our monoclonal and polyclonal services, mono meaning one. So what we do is we usually start with a mouse, a rat, a hamster, and we will, again, raise that antibody by immunizing the animal. Then at that point in time, we harvest the spleen. From the spleen, we then select cells that seem to be responding, and we fuse them to cancer cells. Those cancer cells then are grown in culture, and they keep reproducing the same antibody over and over again, and you can produce large quantities. We offer vivarium services. Our facilities, our animal facilities, are very complex. They're designed to handle multiple different types of species, but yet have flexibility. Depending, we work with transgenic animals. We work with large transgenic animals such as cattle, goats, and sheep, all the way down to mice, rats, and rabbits. And we'll breed them for the clients, produce these, trying to keep the strain clean, as well as supply certain services to be done with those animals as well. The antibodies that we produce need further processing going downhill. So we do the purification, conjugation, and prepare them to be used directly in an assay development format, as well as our contract manufacturing, where we're actually going and filling different types of vessels, labeling them, and preparing them to go directly into the diagnostic kit. You can see a wide variety of different species that we've talked about here. The diversification in these species, as you know, they're all different. They require different types of housing and supplies. So when we're working with these animals, it's important in our training, our programs with our technicians, that we teach them that. And we approach it in a direction of, in other words, as you might hear of a student being taught Spanish or German, we're teaching them to speak donkey, speak rooster, speak sheep, so that they understand how these animals communicate, how they do. It gets down to a big part of husbandry. Sheep all coming up to a feed trough together. A sheep might come up and stand side by side another sheep. And if you're not looking to see if that sheep's eating, or you don't know how to read through its condition through its wool, to see a spiny back or something like that, you might miss that an animal is not doing well. We teach them to look for, say, wool fever and other issues. Other species, we look at discharges, we look at different things. We have our own systems where we're doing hematocrits on the animals. We look at their hematology reports, we look at their protein levels, and there's a lot of control that the typical farmer would not go through when working with the animals. But it's very, very important to us that our technicians understand this and learn the science of each species and the uniqueness of the animals. Another part that goes along with this is animal enrichment. So as they learn about the animals and their personalities, we ask them to develop different types of toys, different types of animal enrichment for these animals, so that they don't get bored being in a pasture or in a cage in a pen. And they've come up with some really great ideas and different toys for the animals. I think, the pigs, one of their favorites is the bowling ball. We used to hang a chain and a small tire in there, and they play with that. So we try to mix it up and make it, unique to the species. But it's really great to get the students, especially when they're doing internships, to make this one of their projects to come up with new toys for the animal enrichment program of our animals. Going to pick just one species. We decided we'd have the cow here just to kind of, again, express what happens with the animal, what are the type of products that can come from it. So we've talked a lot about the blood. Here again, we talk about coagulation testing to see how fast you've just been in a car accident, you're rushed to the emergency room. Before they can start work on you, they want to know, do you need a transfusion? Do you need plasma because you've lost a lot of blood? So they'll do a clotting test right at bedside. And that test is made from bovine plasma. And there'll be other things. Maybe they're going to glue you back together again because it's sensitive tissue area rather than suturing you together. So there's things called fibrin glue. And they'll use the fibrinogen and thrombin from the blood of a cow, mix the two together to make this glue to glue people back together. Different other products that you see there, the albumins and other serum proteins, again, are used in the development of different types of diagnostics. We talked a little bit about the collagen from the hides, bone and connective tissue, bone powder used, as you probably know, in dentistry, and also bone repair. The different types of glands and organs are used in multiple different types of research, as well as the eyes and other programs. So there's a lot just from this one animal. You can see all the different types of products that can come from it. So we have both donor herds where we have the animals that are raised on the farm or kept on the farm as blood donors, serum donors, and then we have other ones that, in other words, we collect products from the abattoir. You want to do some talking here? Okay. We'll mix it up here.
So some of the products derived from blood, of course, you draw the blood out of an animal or a person, you can separate it out into its different components. On the right side, you can see that you have plasma, white blood cells, and red blood cells, and the percentages they typically account for in a normal species or a normal animal. Your plasma serum is about 55% of the total volume, and is usually a golden or a honey brown color. And it's mostly your water component. It has most of your enzymes, salts, nutrients in it, as well as your protein. The red cells, you bring the air to the different organs of the body. The oxygen itself, the hemoglobin, they have no nucleus. So they are something that we typically, if we're manufacturing serum for antibodies, we don't utilize the red blood cells. They form a clot and that's disposed of. In other cases, like Greg mentioned, where you're testing a hematology analyzer, red blood cells are very important. Those red blood cells are what that hematology analyzer is going to read. So if you go to a hospital to see if you're anemic or see if you have any number of blood diseases that impact the cell size, they're going to want cells, red cells from a variety of different species to make sure that that instrument is working appropriately. So you might take blood from a rooster, which has a small cell size, or a goat that has a little bit bigger, all the way up to an alligator. And that alligator has a huge red cell size, but it's a known cell size, it's a defined cell size, and can tell you whether or not your instrument's working properly before you even put a human red blood cell through it. So then you also have your white blood cells, which is where, of course, all of your infection fighting comes from. They're your immune cells, and they actually have DNA in them. There are also platelets that go through and help in the clotting, as well as a number of other components in the white cells that go through and just help your immune system overall.
One of the things interesting about bovine blood, horse blood, equine blood, is that if you were just to go to bleed a horse, that diagram that you saw in there, that tube with the different colors in it, the blood would do that naturally. But if you went and tried to pour that blood out of that container, it wouldn't come out. Or if it did come out, it would come out all as one piece, as a gel. Because the fibrinogen and thrombin in that situation is not being tied up by the red cells. Typically, when you want to separate out the serum, you do it by what's called a centrifuge. By centrifuging out, that pressure of the down cervical force pulls the red cells, which will hold back your thrombin and fibrinogen. In horse blood, the red cells settle out naturally and leaving the fibrinogen and thrombin up in the solution. So it turns into basically like a gel. Just as a little side note. Sorry, Gary.
So one of the machines that we just briefly talked about, just not naming it, is a Coulter counter. This is something that's utilized in a number of hospitals, and it does detect anemia as well as many other blood diseases. They use and purchase material from us, whole blood, from roosters, goats, geese, and alligators. We don't have alligators on site, but we help develop the method to bleed the alligators. And that material is combined together to form what they call a calibrator. That calibrator is what's put into the instrument to make sure it functions as expected when the machine's being made and manufactured, as well as when it's in service out in the field itself in the hospitals. So a number of hospitals have these for their diagnostic testing that goes on, and it provides you one application of red blood cells. As Greg mentioned, another one that's easy to, we deal with on routine basis is sheep red blood. That red blood goes to a company that tests analyzers to detect whether or not you are in a state of, when you're in an operating room, to make sure you clot. It detects your clotting time. And if you're clotting too fast, they thin your blood when they're operating on you. If you're not clotting fast enough, they want to try and make sure that you are clotting, or you might need a transfusion because your blood is coming out so fast. So they test that material with sheep blood. There's a lot of known issues with human blood that people don't want to use that for testing. There's so many regulations around human blood. So what they've looked at is what blood mimics human blood in their process as much as possible. And then they come to us. And that's why we have so many different species. It's not just about the antibodies. Sometimes it's about the things that Greg mentioned with the horse. It's the different properties of their blood. And we have to understand that and help them through it, because sometimes not a lot of people know all the different properties of the blood. We're an expert in this area. So they'll come to us and say, well, what can I get the most blood out of that mimics a human in this instance? And we might say, well, that's a pig. And then another client will come and say, well, I'd like to do this. And we'll say, well, that's probably going to be a sheep. So it just depends on what they're looking for and what we can provide at that time. So your serum actually contains antibodies and a number of other proteins. So we're going to talk about the antibodies a little bit more over the next couple of slides. They're a protein that's produced during an immune response. So anything that's foreign to your body is considered an antigen. You make an antibody to that antigen, at which time then you will continue to produce that for after every exposure. So this is why vaccines work. You get a vaccination, go to the doctor, you get your shot. It's typically a killed or an inactivated material. You develop the antibody so that hopefully the next time you see it, you don't get as sick as you would after the first exposure has already occurred. So they can recognize, bind to, and neutralize a pathogen or any number of foreign substances. And you can have billions of different antibodies. Hopefully it prevents you from getting the flu when you get your flu shot. You hear some randomness sometimes, oh, how effective the flu shot is this year. Well, they're basing it off of what they're seeing coming out of China. But if they don't pick the right strains, it'll still work, but it just won't work as well. Hopefully you don't get as sick if you would have had the flu shot, but if you didn't get it, you're guaranteed to get sick. So when you get the flu, it could be drastic, and that's why they do recommend as you age to get the flu shot. So this is a diagram of an antibody. And again, we talked about an antigen. That is something foreign to the body itself. Could be bacterium, viruses, parasites, pollen, food proteins. It could be a drug. One of the benefits of your drugs normally is that they are not immunogenic, and that's why they work. But if they are bound to larger proteins, you can actually make an antibody to a drug. That's what they use in clinical development to determine whether or not a drug's effective as well. So it has become part of our expertise, as Greg mentioned, to make antibodies to these small molecules that the body doesn't even see as foreign. So the immune cells generate antibodies that are highly specific to a particular antigen. If you have an antibody against the flu, it probably won't react against a different antigen. It more than most likely won't have much in the way of cross-reactivity at all, so that you have to have a new set of antibodies made against these other antigens that you've been exposed to. And that's why you end up having billions of antibodies in your system. So how do you make antibodies? Well, we talked about the doctor model, and that's basically what Lampire does. So we work with our clients, or our clients come to us with an antigen. We help them design and make the antigen from scratch. It could be a protein that they know about, and they only want a small section of it engineered, or it could be a drug or any number of other things. So once we have the antigen, we actually inject our animals. Same way you go to the doctor and get a vaccination is exactly the same thing we do. Then over time, they make the antibodies for us. Then every month they become a blood donor. Just like you have the ability to go donate blood, there's a minimum weight requirement for you to do so. I believe it's 90 pounds, and you can donate 1 pint of blood a month. So our animals are very much so the same. So the smaller animals can't donate as much blood to us each month, but the large animals can. A cow can typically give you anywhere between 20 to 24 liters a month, or equating to about 6 gallons of whole blood a month. So if you need a lot of antibody, of course, you would select a larger animal. Your research model animals are typically your rabbits. Your production animals are your goat, your sheep, your donkeys, and your horse. And they get to live out their life basically getting 1 injection a month and bled once a month. And they get to go out in the field and eat and come in for those 2 occasions each month and make us product that we can utilize and test any number of diseases. We can test for diabetes. We can test for drugs of abuse, for pre-employment screening. We can test for pregnancy and beta HCG. So some of those tests that we just talked about, it can be for diseases, conditions, identification. Do you have an allergy? We can tell you that by using an antibody to determine that. Illegal drugs and even antibiotics. So how do some of them work? Well, I think most people are familiar with a pregnancy test and the operating principle here. So there's a piece of what looks like filter paper in the pregnancy test itself. You can see the analyte on the left. That contains the material we're trying to detect. In the case of pregnancy, it's a hormone called beta human chorionic gonadotropin, or beta HCG. What we've made is the antibodies that are conjugated to the gold nanoparticles. So those antibodies we've made, someone takes and conjugates them and puts them on the strip. So by adding liquid to that strip, it flows from left to right. And you can see what's called a test line of the antibody and a control line of the anti-IgG antibody. So the control line is always going to work because it's detecting something very specific that everybody has. That's usually what gives you the negative sign on a test kit. Then you have your test line, and if the material is present, and you can see how it works, the analyte is there, it combines with the antibody on the gold nanoparticle, then it gets captured on that test line. Well, that line lights up, and that then tells you you're pregnant. So some people have, they have all these different novelty ways of detecting if you're pregnant. It's a plus sign. Well, one of those is your control line. The other one's your test line. So if it's plus, it just lit up your actual positive test line. Whereas your control line always lights up, and that's always your negative line, where it could be 2 lines instead of 1 line and you're pregnant. So that's the methodology. We make those antibodies for each of those different steps. It could be the antibody that's on the gold nanoparticle. It could be the antibody that's on the test line. It could be the antibody that's on the control line. And that's what Lampire does. You wouldn't know us because we're not on the test kit. Some of the newer methods that are coming out are actually to use antibodies to target cancer cells. There's a number of immunotherapies being discussed right now where they're actually putting drugs on antibodies, and they're able to utilize them to go at and target specific cancers to actually reduce their efficacy and growth. And that's been really novel at this point. They're seeing very good results with it, and they're trying to determine if there's other ways of making the antibodies available for different treatments also.
What they'll do is sometimes make this monoclonal antibody that we told you about, and they'll fluorescent tag it, inject it into the patient. The monoclonal antibody was developed against the patient's own cancer. This way here, they can find where the cancer is spread to in the body. Then the next step, what they'll try and do is attach the chemotherapy to that monoclonal antibody, which was made against the patient's own cancer, so that that monoclonal antibody will go directly to where the cancer is, and not, in other words, go to areas where it might do damage to other organs and tissues.
So some of the additional research that Greg just talked about was that combination of the antibodies with fluorescent molecules or enzymes, and in targeting specific disease states or disease molecules. You can actually see anything then. You know, we're able to actually look at an X-ray or a CAT scan or something of the sort. If they're fluorescing, it'll be picked up and we can tell exactly where the cancer is throughout the body. And it can almost target anything as well. As long as we can make an antigen that some animal sees as foreign, we can make an antibody against it. So of course, we've been doing quite a bit recently against COVID. That's the hot topic most people are talking about overall. Viral transport media has been a big sales point for us. The applications across the board where we're making antibodies targeting this material, you know, whether they're in rabbit or chickens or different reagents to even just make the kits, selling bovine serum albumin as a blocking reagent in the detection kits. You know, we're not a pharmaceutical, we're not coming up with treatments, but we want to help try to determine if you have that material or if you have that disease, and then hopefully point you in the right direction for treatment. So of course, you know, what are the prospects going into the future? You know, life, physical, and social science occupations, you know, this is always a growing job sector. I don't think since I've been involved with it, and it's been 23, 25 years now at this point, I always think of these as high priority occupations. The biotech industry is notorious for high priority occupations and continuing to grow. Everyone talks STEM, but you know, a lot of this work comes into play with animals. Almost all of it starts with an animal in the biotech industry. That's why it's called biotech, right? You know, the median annual wage for life, physical, and social science occupations was $68,000 in May. This was higher than the median wage for all other occupations at $40,000 basically. So working with animals, we have a couple summaries for you to look at. You know, vet technologists and technicians, some people that do work with us. You know, the work environment, you can see that on the screen. You know, they have 2-year or 4-year degrees. You know, their median annual wage for vet techs starting is $35,320. And job outlook, of course, employment of vet techs and technicians is projected to go from 16% from 2019 to 2029. That's phenomenal. And you know, companion animals and everything else that's going on in the world right now, you know, how many dog shelters and cat shelters have you heard that are out of animals because people can't go anywhere? So they've adopted quite a bit there. So you're going to see an increase in the vet techs and technicians. Then you have your vet assistants and laboratory animal caretakers. You know, this is something where it's a little bit more of a controlled environment, a lot more regulation to it, like Greg had mentioned. You can see the median pay there is about $28,590, if not a little bit more in 2019. And that's projected to grow about 16% as well. So on the farm, agricultural workers maintaining crops and tending to livestock, we are as much as possible self-sufficient with regards to the crops and what we feed our animals. You know, we have our own crop manager. We have people that get on the back of the tractor and go around and make product for us. You know, just starting out about $25,840, but the growth at this point isn't as high as some of the other ones. It's only about 1% from 2019 to 2029. Biological technicians, of course, you're typically in a lab, but could be out in the field working with animals or looking at the animal to obtain the blood. We utilize them in both situations, typically somewhere around a $45,000 median annual wage, and job outlook's about 5% growth over the next 10 years. Microbiologists, of course, this is a little bit more structure, anywhere from entry level all the way up to PhD. Some of the testing that we do on our blood requires microbial content testing, and that can be upwards of $75,000 for a median annual wage, and it's a growth of about 3% over the next 10 years. Chemists and material scientists, we utilize chemists and material scientists to extract the actual components from the blood. So as Greg mentioned earlier about purification and conjugation and digestion of our antibodies, anywhere from a bachelor's degree to a PhD again, and the median annual was about 77 or 97,000 for a material scientist. And they're expected to grow about 5% over the next 10 years as well. So some of the skills that we look for that are fundamental and ag science related. Fundamental, you've probably heard them all at some point in time. Good communication skills, be a hard worker, passion for what you're doing, attention to detail, get involved in what you do, ability to work on a team, problem solving. One of the things that, we just talked about this morning, we do a lot of training in-house, and realistically, one of the fundamental skills that goes as part of that is your willingness to learn. It's not always about the training. We can train you, but if you don't want to learn it, it doesn't matter. So you really want to have that person that's enthusiastic for what they're doing, and they really want to learn and understand what we're talking about. Of course, ag science related professional skills, there's math and algebra involved. I know people don't always think about that, but that is the case. Basic understanding of chemical safety is a bonus. Scientific method, animal husbandry, crop management, phlebotomy techniques. We get a number of phlebotomists that come to us and are looking for positions as well, rather than, they learned early on that they don't want to deal with people. Started out, thought it was a good job, got involved with it, and then found out that, going into the hospital and working with people just wasn't their cup of tea. So we ended up with a bunch of phlebotomists on staff as well. So some of the job opportunities overall with us or throughout the industry, animal technicians. We utilize a variety of animal technicians from a high school diploma through BS degree to maintain our animal herds. We have, as mentioned before, anywhere between 5,000 to 7,000 animals and 20 different species. We have animals that people come and talk to us about because by now we're probably the experts in them. There's not many people that keep emu herds. So if someone wants to know about an emu, they probably contact us and want to find out, how do we handle that? That's one of the things too that we try to get people to understand is that we've dealt with a wide variety of species. It's not just your normal domestic animals at this point. We have opportunities in the lab, manufacturing, purification, monoclonal. You can see customer service. Quality control, quality assurance is one thing that Greg mentioned is usually short-shifted in understanding, and it's mostly a paperwork aspect, but there are people that want to work in lab situations or be involved with the sciences that they don't want to actually be in the lab or don't want to handle the animal, but want to have that passion and have that drive to actually do something in the life science industry because they want to help mankind. They want to have that, I did this today and I did the best job I can do today, and I'm out there helping people. My company does all this cancer research, or we make antibodies that diagnose all these different tests that are out there. That's a good aspect too, and people don't always see that. They think when you're coming through for biology or something else of the sort, it's all about the lab. And of course, some more of your competitive skills as you would progress. If you go for your master's degree, your PhD, your veterinarians, and what we expect of them when they come in.
Right. One of the things I'll interject here again, is that a lot of the students that go through tech programs, okay, in days of old when it was called VOTech and things like that, it was thought that that was the end, that a student went there for training to learn a skill and then, in other words, went into the workforce. Today, there's so many more opportunities, and it's important that we get the counselors, the parents, and students and teachers all on board to that opportunity. One of our success stories in Bedford County was that one of the students early on in the program went through all the way through vet school and is now a practicing veterinarian in the Bedford area. This is a true success story, and what it is, and it's why you do what you do. If you're passionate about what you do, if you enjoy what you're doing, you're going to be successful at it. And that message is very, very important to get through to the students. Find out what it is that you like and what you want to do, and follow that. I never knew, in other words, if you heard the story of how Lampire started, it was started in between my sophomore and junior year in college, and I showed up the wrong place for a job interview at the wrong farm, and the rest was history. And now I'm part of this great organization producing products that are helping people around the world. And what we tell our employees every day when you show up for work, not only are you learning and earning money and moving forward, but you're helping human lives, animal lives. You're helping people all around the world. And nothing could make them more proud and take ownership of that. When our clients come to audit us to make sure that we're following procedures of government agencies, we ask them to talk to our staff at all different levels to explain the products that we're producing for them, how they're being used. So as you look at these job opportunities and graduate degrees, what the opportunities are, it should be made to know to students that this is real. This is what you can go to. And one of the things that's really nice about this industry is that a lot of companies like Lampire and some of the bigger biotech and pharmaceutical companies offer tuition reimbursement. So here they can graduate from the high school curriculum, go into work for the life sciences, and possibly, if they have a desire to go up the ladder, there will be a tuition reimbursement program for the students to progress and go to it. And they should be encouraged about that. But we need to let the counselors and the parents know about these opportunities as well, because there's still a stigmatism out there about these tech programs that it is just, an end of a means, and it's not. It's the start of a great future.
So some of the industry trends and characteristics, of course, we're seeing the industry move to more antibody engineering, recombinant antibodies, individualized medicine, like Greg talked about with cancer treatments, the treatment plans, automation of processes, animal-free medias, DNA therapies. It still involves the animal to some degree. And then, of course, your characteristics of what we look through for people coming on to join us: hard working, good attitude, enthusiastic, practical intelligence. You can be book smart, but if you can't apply it and understand how it looks in the field, it's a different story. Quick learner, passionate, team player, get involved, participate. Don't sit there like a bump on the log. Be conscientious and be accountable for your work. So I believe now, if you have any questions for us.
Open up to the group. Any questions, thoughts, ideas? Did we touch upon what you were interested in? Did you learn anything? John, are they muted?
Give everybody a few seconds there. I just promoted everybody to panelists so that they can share video if they so choose and unmute themselves. A couple familiar faces, at least to Greg, that you might see on here. There's George from Upper Bucks.
Right.
Yeah, so folks, now's your time. Lampire is truly a hidden gem here in Pennsylvania. Typically the food processing industries and, big ag names that are around, that's what you hear about when you think about Pennsylvania agriculture. But Lampire, as you just saw and heard, embodies the agricultural biotech industry here in Pennsylvania. And they are just a wealth and treasure trove of expertise, experience, guidance, advice. And I'm going to bet that not many of you even knew that they were here. So we look to change that. We have developed a fairly strong partnership with Lampire, and we plan to continue engaging and allowing you to engage with them. So this will be the first of what we hope to be many touch points and moments of interaction that we provide for you with Lampire. But if anybody has any immediate questions, comments, responses to any and all that you just saw and heard, now is your time. We have a few minutes of scheduled time. I don't know if Gary and Greg can hang on for a few minutes if we run past, but any questions, any comments, anything that jumped out at you that you never thought of or never even imagined possible in the ag industry before?
So I guess I can start. Hello, thank you gentlemen so much for your presentation. My name is Darla. I teach at Cumberland Valley High School, about 10 minutes south of Harrisburg. The thing that jumped out to me that I just maybe want some of like your input on was just all the different applications for like that you can get from like the blood products, the serum, etc. So I guess my question is, if I had to pick one of the topics that you discussed today to integrate into my animal science class, which is generally for sophomores and juniors, what topic would be the most prevalent that would prepare them and maybe get them interested in biotechnology?
You might have heard me say during the presentation and it's maybe, you don't think about it as being directly animal related, but I'm really trying to encourage these courses to look at the quality program, the quality of the feed that they're feeding the animals. What is that established of? When they go to the feed mill, maybe for a visit, what do they want to look at there? What are the important characteristics of that feed mill? And again, as they learn, there'll be certain standard principles in the food industry that goes by side. Mr. Dietrich, you know that. In other words, you've been exposed to the food. There's a lot of correlation between the animal industry of quality and the food. One can't exist without the other, both having good quality programs. And I think it gives the students a different opportunity to look at that as quality as a career, and it's needed. If you were to go and look in the life sciences and see what field is never filled, it's the quality side. There's never enough, it sounds funny, but there's never enough quality people. And, you have quality control, you have quality assurance, but regulatory is so important. And in every organization, I don't care if you have a dairy farm at home, you want to look at your quality operation and how you can improve upon that. Whether what you're looking at in a dairy farm, when you're looking at the milk and that. But in other words, it's just like a computer, right? Garbage in, garbage out. The same thing, the feed that's going into the animals, whether it's poultry, swine, dairy, beef. In other words, better quality products going into those animals to try and help create efficiency in that. And what are we trying to produce for the table? Better carcass, better meat, better loss ratio. And, if you think about, way before you guys maybe got into agriculture, and you start thinking about the different types of breeds of animals. At one time, we had these box cars, right? If you looked at livestock judging, what it was back maybe in the 50s to 60s, they had these little boxy animals. Then we decided, hey, we needed large frames, so they brought these big bony animals in, right? And then the industry found out we're paying high price for bone, and we're not getting as much meat, and so the industry kept evolving it. And believe it or not, that's all about quality. And your breeding programs and genetics and all this other stuff. So if you were to ask me that question, I would say this, because if for some reason they find out, I don't want to go back to the farm, I want to go to something, they can go to any industry they want if they have an understanding of quality.
Yeah, great question, Darla, and great response and advice. Any others? If you do, please use the raise hand icon so your name jumps up to the top of the list and we know that you have something you'd like to contribute or ask. Don't be shy. So I'll just add on while maybe somebody's thinking of a question or a comment. Those of you that are in our Breaking Down Biotech professional development program, we have our very first, and unfortunately at this point only in-person touch point coming up this weekend on the biotech basics. Hopefully this was a good primer. I think it was a great primer to get you excited about and sort of setting the tone and the stage for what this year of biotechnology professional development has to offer and the industry that awaits the students that you're going to take this information and apply it with. So this was a great precursor. We're going to sort of dive down into the weeds a little bit deeper this weekend with Dr. David Garby, who's going to actually have you start getting your feet wet with some of these biotechnology activities and labs that Gary and Greg and Lampire utilize frequently. Any other questions? We're 3 minutes past our scheduled hour. We do hope to, unfortunately, COVID, hopefully Lampire helps develop the cure and the vaccine right quick so we can get you back out into the field and do larger group events because if at all possible, we would like to facilitate and get you to tour one of Lampire's facilities. Right. At some point. It would be great to do it during this biotechnology professional development year. But with all things considered, I don't know if that's going to be able to happen or not. But so it's not the last time you're going to see these two great guys. And I know that they are willing to help out, and their track record of helping out education, specifically our ag ed programs, speaks for itself. So any other last minute comments, questions before we adjourn here today?
John, maybe you can share our contact information with the group. If they have any questions, they can reach out to us at a later date. And Mr. Dietrich, we're working on your wish list there. We should have some things for you. We're excited to see it and very excited to take the tour of your new classroom. Thank you for letting us be a part of that. Well, thank you, everybody.
Okay. No, thank you both, Gary and Greg. On behalf of everybody, I know everybody appreciates it, but thank you very much. Your time and commitment is greatly appreciated, and we look forward to interacting with you more.
Great.
Thank you.
Thank you to everybody who logged on today.